Hello, everyone, and welcome to the fifth and last episode of the Live from the Lab series. I'm Milena. I'm doing my PhD at the University of Exeter, and I organize this event with Siri Kellner from the Berlin Museum of Natural History. Today, I will be your main moderator, and Siri will be one of the speakers. In these 5 lectures, our colleagues and us from the EvoCell Marie Curie network presented bits of our work, focusing on how our research contributes to answering the mysteries of what animal ancestors looked like. And you've seen this in the trailer. So one important concept that we work with is that animals are very different, but they're made of the same cells. And while we can't know for sure what animal ancestors looked like, we can tell that they must have been made from the same cells as us today. And over the course of the series, we looked at animal ancestors from different perspectives, so from fossils to single cell research. And we hope that you learned something new about animal ancestors from that, because today we're going to be doing something a little bit different. So as every time, we will start off with around 20 minutes of talks, and the rest of the time is dedicated to a discussion where all questions are welcome. The topic is the cyber age of science. So first you will hear from my co-organizer Siri, whose research is concerned with the ways of representation of digital data. Then we go over to Kevin, who is generating said digital data and working with animal genomes. So he will talk a bit about why he's doing that, why that's important, but he'll also show you how you can become a part of the scientific process and explore some massive data sets. And our last speaker is Konstantinos, who is a bit of an outlier in the network. So he will show us some of his work on cancer cells and discuss the intersections of fundamental and applied science and why one cannot exist without the other. And yeah, that's been enough intro. With that said, let's open for the talks. And Siri, do you want to start?
Yeah, with that, I like to continue I will start my talk. So my name is Siri, and I was born in Norway and grew up in Switzerland. And at the moment, I'm doing my PhD project at the Natural History Museum in Berlin. And today I'll be talking to you about the changes in scientific practices over time and how we could exhibit the science today. So an important thing that all scientists who are presenting in this series have in common is the practice of comparing objects in their research. And comparing objects is important for any study of life, but also in the fields of geology, cosmology, art history, and many other disciplines. And this comparative aspect in research is connected to collections. If you think about collections, you might imagine a museum's collection, just as the one as I'm sitting in right now. I'm surrounded by millions of physical objects that were collected over hundreds of years. And in the field of natural sciences, the practice of collecting physical objects of nature was essential for generating knowledge from the 15th up until the beginning of the 20th century. And for that, expeditions around the world, many of which were colonial, brought vast amounts of objects back to Europe during that time. And collectors and scientists were trying to find order among these collected objects. They were trying to make sense of them. From around the mid-18th century, big collections began to be established. So, for example, at the University of Berlin, the Natural History Museum in Paris, or at the British Museum in London, that you can see in my background now. Yeah, just to name a few. And so this engraving behind of me of the British Museum in London was done in the mid-19th century. And so during that time, many of the natural history museums became overloaded by objects from these expeditions. And they were still trying to order these objects to make more sense of them. Around that time, in 1837, Charles Darwin did his first drawing of a tree diagram representing the relationship between animals. It was the first time when he was visually representing the idea of evolution scientifically through a tree. And the theory of evolution, however, got first presented and published around 20 years later by Charles Darwin, as well as Alfred Russel Wallace. And this was mainly made possible through the practice of collecting and comparing objects systematically. So let's try to make this even more clear. So if you have only one object, such as this one, it's a helmet sea urchin, a skeleton of a helmet sea urchin, we can't tell really much about its past nor its future. Why does it look the way it does? How did it evolve, and what implications might this have for its future? But if we take several objects, so with this echinoderm as well, and then we take a coral as well with and we look at another coral. This one is from China, I think. So if we have several objects, we can start to compare them and group them. And we can tell, well, these two objects are probably more related to each other than this one. And yeah, so and if we have more objects from different time periods, that's even better. But to group these objects the best way possible, we can use scientific methods. So what natural historians did and still do is to collect and compare these objects and analyze them, so make a detailed examination of them, and from that generate knowledge. And today, the EvoCell network and other scientists studying modern experimental biology do exactly the same. They collect, compare, analyze, and generate knowledge to answer evolutionary questions, but on a much smaller scale. And the difference is the study objects are not really visible to the naked eye. They're cells and tiny molecules such as the DNA. And the collections they use are digital. They're comparing digital data and visualizations. So in my background, you can now see some examples of the kind of digital objects they compare and examine. On my right, you see microscopic images, genome sequences, which Kevin will talk more about in the middle part. And on my left, you can see microscopic images where a specific gene is activated and visualized. So working with digital objects is very typical for modern research. The objects of interest are either very tiny, very big, or very distant, and which makes which means that they need to be made visible through the use of technology. And throughout history, researchers have continuously asked new questions about the visible world that surrounds them. And this led them to the boundaries of visibility and making sense of the world. New scientific technologies and methods were developed to cross these boundaries, enabling us to generate images and data like the ones you can see behind me. They make the invisible visible, and they help us to make sense of the world. And this development makes the research also much more abstract, and I would say makes it seem more complex than it actually is. With our ability to enter a previously invisible world, science has also become much more specialized. And because of this, individual research projects often get lost in communication. So for example, if you go see a natural history museum exhibition or science exhibition, any other things, you often don't really see the behind the scenes research that is conducted, or you hardly learn how you can explore and research these kind of digital objects yourself. And this brings us to my research. I am currently studying how to exhibit specialized science that is not immediately visible in cyberspace. And I think cyberspace has many different advantages over the physical space, like having an infinite space to exhibit the tiny aspects of research. And we can create as many layers in an exhibition as we want in cyberspace. Furthermore, it's less authoritative and more participatory. And the online world can be accessed from anywhere with a computer and internet connection. And most importantly, I think cyberspace is naturally the most suitable environment to exhibit digital images and data, which visualizes the invisible world. It is a tool that can transform the information into objects and connect them in a system. And for me personally, exhibiting specialized research in cyberspace or anywhere is not about making individual research projects super visible in the media. It's making them accessible and with that sustainable. And especially today, where it's increasingly difficult to distinguish between real facts and misinformation on the internet, we need reliable resources. We need to give everyone access to scientific evidence and to the tools that enable us to make sense of the world. And I think as scientists, we have the responsibility to make research as accessible. And there's already a lot of data and images and research findings online for free, theoretically accessible, but practically accessible, not so much. And making specialized science visible and accessible can be done in many different ways. I am looking at how to exhibit them and open up a space to explore these objects and relate them and make sense of them ourselves. And I think we can learn a lot about how to exhibit in cyberspace in a meaningful way from museums and their exhibitions, so as not to follow the common nature of most online platforms where search algorithms and ranking systems dominate how we see this non-physical space. An online exhibition should be a space for self-driven education, a space that not only answers your questions like a Google search, but raises more questions and increases your curiosity. And with that final thought, I will pass you now over to Kevin, who will share more on his research in the cyber age. Thank you for listening.
Okay, good. So you can hear me okay, and you can see my screen, I hope. It is the range of biodiversity that we must care for, the whole thing, rather than just one or two. This is a quote that I really love from Sir David Attenborough, or at least accredited to Sir David Attenborough, that emphasizes the need for humanity or mankind to really care for and look for and appreciate the biodiversity that exists on planet Earth while we have it. And this biodiversity is rich. It comes in vast amounts and types, and also poses a challenge that the EvoCell network, for instance, has attempted to face, which is explore the many different types of organisms that exist as best as we can. And we've had this from the numerous talks over the last few weeks from my fellow colleagues. And not only that, but as you explore and analyze this data, then you actually are generating information. And so how do we make sense of this? And so this is a challenge in the current age that will continue plaguing us in the future. And how to face it is quite interesting, but also daunting. One such challenge is the capacity for us scientists to actually profile the DNA that exists across all life forms. So here I highlight a story from the Earth Biogenome Project, where their aim is to essentially analyze the DNA of all living organisms, with the exception of bacteria and viruses. Not that those are not important, but other groups are also working hard at that. And this schematic hopefully illustrates how daunting this is, right? To try and infer relationships across all life forms is quite challenging. And it's not just the scientists at work, but also all of us who are paying taxes, of course, that contribute to this effort. And so it's really a community effort, indirectly as well as directly. And my contribution to not the Earth Biogenome Project, but the project, hopefully, of sampling biodiversity on Earth, comes from this very beautiful animal, Dumeril's clamworm. This is the same animal, actually, that we had from Milena yesterday, who's also studying them. And a number of labs are interested in this animal for a variety of reasons, their development and their biology. And I find it fascinating that we can, of course, analyze the DNA, but also offer this as a tool for the community to use, which is something that Costa will also briefly mention. And what is also, at least for me, what was super fascinating about working on the DNA of this singular species is that it's quite similar to many other DNA that we actually have explored thus far. So this is actually a snapshot of the Dumeril's clam worm genome. It's only a small fraction of it, but it's essentially a string of chemical compounds, A, T, C, and G. And somehow from this very, what looks like a boring schematic of just letters, we are able to then build proteins, DNA, genes that can be expressed in specific cells. And then this set of sequences also adapts to the environment over time, a process through which we call evolution. And somehow making sense of all of this is already daunting at the singular specimen level. Now imagine us looking at millions of species. And this is quite challenging. And to even analyze such a data set, which is something that I'm involved in, we need supercomputers, and we need to be able to not only analyze it, but store the information and work with this. And now I want to steal a moment of your time and show you some of the supercomputers that we have that I'm working with. And I'll just quickly escape this room for a moment. So I'm currently in EMBO, the institute I'm working with. And hopefully you can see, this is one big room, and each of those panels consists of thousands of computers, central processing units or core processing units. And this room spans from here all the way to the other side. And if I can quickly show you, this is how deep the room is as well. So thousands and thousands of computers. I think in EMBO, we think we have 14,000 processors. And somehow from all of this, we have to generate data, analyze it, and then compare across many different organisms. And I will stop sharing. Which then adds to another challenge, which I'll briefly mention, which is given we're generating so much data, how... Firstly, I'd like to apologize for the technical issues that I had during the live talk. And so I just wanted to finish off what I was about to say, which was, despite having so much computational infrastructure to analyze a lot of data, one of the challenges that remain is how do we then actually use this data and browse it in a user-friendly way, a way in which allows all of us to access the information, just like we would if we were to go into a museum, like Siri said, but also to actually learn from it. And something that's dear to me, at least in regards to analyzing genomes, there's plenty of resources available that allow us to visualize genomes or DNA sequences. And often these are hosted in data portals known as genome browsers. And this allows you to essentially navigate your organism of interest, genome or DNA, look at specific proteins or genes that you're interested in. However, it's still limited because it's mostly catered for specialists who can actually use this data. While it is publicly available, I think it needs quite a lot of learning to know how to actually access the relevant information of interest. I wanted to quickly then show an example that I think is a success and is probably looking towards this is where the future is heading. And that is in reference to the nervous system. The nervous system is, of course, another highly complex data set with many, many cells interconnected. And so how do we actually understand the brain without appreciating the structure? And so people have a lot of data actually accessing the structure of the brain, but then to analyze it becomes a whole, it's basically, it's an infinite problem. And to help kind of reduce this problem, scientists have now offered games where you can actually, anyone can go into this website and play the game of tracing, or essentially manually reconstructing the nervous system. And this is fascinating because in a world where we're interconnected, and in a world where I think we need to be more appreciative of each other, so not just specialists and non-specialists, but all of us, to engage actively in doing science that is relevant for all of us. And this will only allow us to then access the huge data sets and the huge biodiversity that exists. And so, for example, here, this game has already been played by over 200,000 people and has a far wider reach, 145 countries. And this is the future that I, at least for myself, hope for in the age of cyber data science, where we can all engage actively in analyzing data sets and appreciating data sets, just like we would in a museum, and to actually learn from it and appreciate the history of science, because then that'll help us understand the science that we need to do, and the science that we should do, and the science that we all do. And this is something that Costa will touch on as well. Thank you for listening.
So for those of you who don't know me already, I'm Konstantinos Geles. I come from Greece, and I'm doing my PhD in Genomics for Life, a spin-off company of the University of Salerno in Italy. In this talk, we would like to concentrate on basic and applied science, how are connected with my project, in which I study how cancer stem cells differentiate and which molecules play an important role in that. First of all, what is basic science? Basic or pure science is the research that people do for the sake of knowledge. Curiosity is the main reason driving humans to gather information about the world. Our biggest desire is to expand our knowledge in various fields and try to understand how life works. Let's take, for example, the case of bioluminescence, which is the production and emission of light by a living organism. Do you remember Clytia, the tiny jellyfish that has a lot of different forms during her life cycle? If not, I suggest you to watch at least the previous episode named The Secret Lives of Sea Creatures. Well, this jellyfish is also bioluminescent. Here we see the adult form of this tiny jellyfish emitting green light under a fluorescent microscope. Magnifying one of the tentacles, we can see clearly emitting a green light. This is bioluminescence. So, which is the connection of bioluminescence with applied science? A lot of scientists studied it and found that a unique type of proteins under specific circumstances can emit light. This category of proteins are called green fluorescent proteins, and they emit green light in various organisms. After this remarkable discovery, scientists studied them and managed to change them. In the end, they could produce almost all the colors of the rainbow with these new green fluorescent proteins. Of course, they didn't stop there. They were able to use these proteins in different kinds of cells in various tissues. And why they did that? Well, if they would like to study the neurons of a worm, it would make everything easier if they were able to see them under a fluorescent microscope emitting a green light, such as in this picture. This discovery opened a whole new world of application, giving us the chance to be more specific and understand better the various structures of a tissue. This is just one example of applied science, which is the practical application of using discoveries from basic science to solve common problems. Applied science is finding answers to questions like how to treat a bacterial infection, how to diagnose a heart stroke early, how to stop a tumor from spreading. And here is where my project starts. How is it related with my project? Let's go back to stem cells. Are the starting cells which differentiate, change to specialized cells like neurons or muscle cells and form the tissues of our organism. When something goes wrong to these specialized tissue cells, they mutate to tumorous cells, and after many mutations, evolve into cancer stem cells. These cells develop to many cancer cell types, causing metastasis by giving rise to new tumors. So, as some of my colleagues in the EvoCell network are studying about the developmental programs used by the stem cells for organism development through the eyes of evolution, I'm studying about how cancer stem cells mutate, invade other organs, and create metastatic tumors. More precisely, I'm focused in some molecules inside these cancer stem cells that have different numbers respect to other normal cells. How is this applied from biology to bioinformatics? We start from a cell culture or a tumor tissue and extract these molecules. We use special equipment to identify their numbers in different cells. Then we import this data to the computer, analyze them, and search for changes in their numbers. At that point, we get the results that are something like this table. In our example, the molecule A has more numbers in cancer stem cells than the normal one. And this is done for all the molecules we can find. And then together with the biologists, we make more research in order to find about the molecules of interest, if they could be potentially used as biomarkers for the identification of cancer in early stages, or find ways to use them for cancer treatment. As a last note, I would like you to remember that basic science is the roots and the trunk of the tree. Applied science is the flowers, which together they give us fruits. Both are linked, and without basic science, applied science wouldn't exist. Thank you. And I would like to ask you to write maybe questions, or if you have any interesting ideas about how basic science research helped any examples in applied science, please write them in the chat.
And thank you to all our speakers. And just to address the technical issues, so we have Kevin back now. You can ask him questions if you want. And both of the chats are supposed to be working now, both on YouTube and on Zoom. So yeah, I would invite everyone to ask questions to our speakers now. And while you're all writing in the chat, I will open the question round. And I would like to start with asking Siri a question. So Siri, now is your time to complain, basically. I want to ask you what have been the biggest challenges of collaborating with scientists to bring their science to life, and what are the biggest challenges in general, not only in your collaborations, and to keep it positive, what are the best parts?
Okay, I think there are, I can just, talk from my experience so far. And well, the challenges are that I guess every scientist is very busy and has other priorities, especially like so making the first priority when you want to make something accessible that you've been researching, of course you want to do it accessible, make it accessible to the scientific community, and through a publication or by sharing data, and less to the general public. And so that is first of all not a priority, and then makes it sometimes difficult to I guess interact and but I think once and I found that really a big blessing now, being part of the Evocell network and being able to try to see how to communicate and exhibit the Evocell research, is that when you interact with anyone in the network, and any scientists I know have so much passion and fascination for their study objects and research questions, and they really ask really interesting questions about the world around them. And yeah, so back to the challenges of that it's sometimes maybe difficult to get it across, is that big challenge is that sharing data and images is because so when you have a publication and you want to, usually the journals, if it's not a non-open access journal, it's sometimes difficult to get the images from scientists, or the scientists don't want to share the images themselves with the public, because either they want to publish it through a journal that then keep the rights of the image and the data, and if it's non-open access, of course that's then a bit critical. But yeah, I have more positive things to say actually, and yeah, I love working with scientists, and yeah, I'm a scientist myself, so.
Thank you for the positivity, and yeah, I think everyone agrees that open access is very important. To continue on that note, we have a question on YouTube that is directed, I guess, to everyone. And that says that this is what challenges have you faced during your research? And I would like to ask Kevin if he's here to start with answering this question, obviously addressing first his horrible internet situation at Temple. How do you work with internet that is that bad? And yeah, if you want to give any final thoughts on your talk that got interrupted, Kevin, please go on.
Hey, I'm sorry to everyone. That was really bad. I promise I tested it, and it just didn't work. So I also lost you, Milena, there because I think internet is dropping. So what was the question? Sorry, I apologize.
Sorry, yeah, it is. What are the biggest challenges you face in your research?
I think sharing the data in a way that is useful for people, and that in itself is hard to define what is useful. But yeah, it makes it hard to know what people actually need or what people want with the data sets that you have, and therefore to format it in a way that, as Siri mentioned, is not just available in media, but a way that people can actually access it. That's already difficult within the specialists field. It becomes even more challenging if we want to share our data with people who are not specialists, whether it's within the scientific sphere or outside it. So I would say that has been a challenge of mine, trying to prepare a resource that people can actually use specifically for the genome that I'm working with.
Yeah, that is a complex issue. Konstantinos, same question.
Okay, about the challenges. Yes, one part is the way that you can share the data. So fortunately, there are public databases that you can use, and a lot of people, as they publish their research, they also publish their data. So for bioinformaticians especially, they can access them and find them easily. But the challenge in that is sometimes the data may be there, but are not annotated correctly. So you have to start searching through the publications and ask, maybe send emails to the scientists that they did the research and produced the data, in order to be sure that the data that you have, the samples that are in this data, are the correct ones, and maybe there are other issues that you don't know. So the most difficult part, I think, is to make everything public and to make it easy and connected, let's say, in a way, in order to be used from everyone. So just not people that have to be specialized and know how to download and how to find these large databases and use this data from different kinds of research.
Thank you. Yeah, I've encountered annotation problems as well. Yeah, bioinformatics has to have a relationship as well with experimental science. And that is sort of related to this question that we have in the Zoom chat, which is from the category of questions, of more of a comment than a question. That says, so there is not an actual barrier in basic and applied science, but each side feeds each other with data through a dynamic relationship. To which I would say yes, but Konstantinos, would you elaborate further?
Yeah, as I have showed from my presentation, basic research is making the first analysis, let's say, in everything. So every scientist makes a question and basic science tries to answer it. When this question can be answered or potentially give an initiative to a theory, then applied science maybe come there, take this theory, use it, and then make more questions in order to answer to the first question that it has been made from the scientists that started the basic science question. For sure, they're interconnected, and basic science has provided most of the information that applied science has now. And a lot of techniques actually have been studied first in basic science and then changed and worked in order to give us applications.
Yeah, thank you for that answer. So we can continue, let's see, with a question from the Zoom chat directed to Siri. And I like that question. So working with digital exhibitions, you must have seen many examples of data visualization. In the last year, we saw everyone trying their best to make complex data understandable with mixed results. What can be done better? Are there any great examples of visualization? So yeah, we have all been contemplating this issue quite thoroughly, because we want to exhibit our data eventually, and Siri is the the brain behind this operation so far. So what do you have to say?
I definitely can't give a perfect answer. We'll first develop the online exhibition next year, but of course I had a look out, and now with since the pandemic started, of course, so many museums and digital exhibitions everywhere. It's kind of a jackpot, so many case studies to look at. And yeah, so I think sometimes a problem is when you look at digital exhibitions and data visualizations, is that maybe they haven't incorporated interaction designer, UX designer. So what is really important and often forgotten, I think, in online exhibitions is to really study the computer-human interaction. So is it usable? Is it understandable? Is it easy to use? Like what many other online platforms use and do, but yeah, so that definitely can be done better. And regarding great examples of visualizations, I don't have a typical data visualization in my mind, but I really love Drew Berry. He does visualize the molecular and microscopic world through animations. He's an Australian animator, and he does amazing visualizations, and he usually uses more time to research, or he uses about the same time, I think, to research for his animations, and then he produces them. So they're really thoroughly researched and usually reflect the state of the art, how we think, or he thinks, you can imagine the microscopic and molecular world. And I'm a big fan of his artworks and animations. Thank you.
Yeah, thank you. Everyone should look up Drew Berry. It is really beautiful animations. And moving on to another question from the Zoom chat that I think everyone can answer. And it says, do you think that it's worth studying pure science if there's still not a way to apply it?
I think it depends on what you mean by apply. So I think something that I find the more I read up about the human history and what we're interested in, at least on the positive side of things, we are very curious people and we like to explore the world. And part of me, this is just my own answer, part of me thinks that an application of science can be just us exploring the world around us and sharing that, what we see, and sharing what we find and our enthusiasm behind that. So for its beauty, I think it can also be super nice. In terms of how we should fund that and how other people feel, I think it really is depending on the type of infrastructure we have. Some people may want that we have to do science that is applied because then it's taxpayer funded and therefore that is a necessary part of the scientific endeavor. So I'm not against that either. I do romanticize though that maybe there's a portion that can be saved for us just sharing things that we learn about the world around us, and hopefully even by just sharing what we know about new species or how different cells do different things, we can appreciate the world around us, and maybe that's already a win, right? If we can look at the world in a different way than before, that in itself is an application, I think. But I think this also would be different on a different day, if I could answer it. So I don't know, maybe Consta or Milena or Siri can also go.
Yeah, I agree with that, and I would add that the taxpayers' money also funds the arts that are sometimes enriching in a way that is analogous to the way that fundamental science is enriching. So I see value in that. And if nobody wants to continue philosophizing about this, I will go back to YouTube questions. Konstantinos, there's one for you, asking when you contact researchers to further question them about resources and stuff, are they mostly happy to share information and help a fellow scientist?
Okay, fortunately, until now, yes. Most of the time, it takes some emails to start collaborating, let's say, and get more information. But most of them are kind enough to let this information and give their data in a way. But okay, there is a chance that you may get some negative answers. That's why I believe that it has to be fundamentally become all the data to be public. So it doesn't matter if someone doesn't want to publish his data because he wants to do maybe more research on them and publish later his research. But from the time you produce the data, you should publish it in order and save it to public databases in order to give the access to everyone. Because when more people work on something and they collaborate, great things happen.
That is so true. A question from the Zoom chat from someone whose name is also Konstantinos. So the question says, do you think that if a bigger number of scientists started working and researching on different species and producing new data about them and the evolution that has occurred on them, we might end up finding answers to our biggest questions, like who our common ancestors were and how they looked like, etc. I swear this is not a planted question from the network. It is a real person asking it who just happens to agree with us. Yeah, who wants to take it?
I can just quickly comment on it. I think we'll never, we can do endless kind of research as much as we want to, but we'll never ever find out exactly how our common ancestors were and how they look like. We'll probably most likely be wrong because there's always going to be holes and gaps and yeah, it's impossible, I think, but we can get closer to the answer. And on the way of answering such a question or trying to find out that we'll get really close maybe, and we'll answer and find out many other very interesting things, especially that brings us then maybe to knowing and understanding us and the world around us, and also about the current biodiversity, about its future. Yeah, I'll leave it. Maybe someone else wants to take it.
I think Kevin also elaborated on this quite nicely in his talk about the importance of diverse models and etc. And I will go back to asking questions myself. So important question for Kevin and for all of us to think about as a society is you've showed all these thousands of supercomputers, and is this sustainable? There are so many, and there are going to be increasing numbers of them as we keep generating bigger and bigger data sets. And what are the sustainability and ethical issues that we will encounter in this expansion?
Yeah, we had a discussion about this, and I still don't have an answer. It is daunting to think that if, in terms of how we source our materials and the amounts of data that we want to store and generate, then it becomes a problem, because if we don't have the resources to maintain that over the long term, then we can ask questions as what's then the utility and how useful is it for us? as humanity. What I do hope is that we can be more aware by us sharing our insights into what we're doing, our data sets, and communicating beyond just the tiny communities or islands that we make for ourselves as scientists. And the hope there is that by engaging with others outside our field, we can have a type of science whereby it's not just a scientist dictating what is important, but we can actually also do a science that is relevant in today's time and age. And so a science that allows us to think of how sustainable is the research we're doing. Can we move to more alternative uses of energy or uses of materials to prevent, some of the atrocities that can happen not only to nature in terms of the environment, but also geopolitical issues that can arise from this. So yeah, my hope is that by discussing it more with people outside us, we can stop thinking of machines, and data, to more diverse input from people worried about or concerned about the ethical issues of our own types of data sets that we gather, and the materials that we source it, and so on and so forth. That's my feeling, but I definitely don't have one answer for that.
Those are good points, I think, and I would encourage everyone listening to look into and research a bit about the different points that Kevin brought up, because they are important and they are a little bit overlooked in light of more pressing issues that we face as a society. But it looks like our audience today is just as ethically minded as we are, which is lovely. And our next question from the Zoom chat is asking, do ethical issues arise from the free use of data? Whoever wants to take it, I think you can all do it.
I can give a comment. I'm definitely not an expert on it, but I think maybe it's a super difficult question, right? So I think everything should be free and accessible, because if it's not free and accessible, it'd be accessible to someone else, and maybe that person then has the power over the data or the objects or something. So we have to talk about the power of having access to something and other people having not access to it. But then, of course, there are ethical issues with having free access to everything, maybe in a way that if someone uses certain types of information and data in a bad way. But I am 100% not an expert on this, but maybe someone else can also share their opinion on that. And yeah, these are my thoughts.
Thank you, Siri. Kevin, Konstantinos, do you want to chime in on this? Or is it too complex of an issue? It is pretty complex.
Yeah, I think ethical issues are there, and I think we're only just beginning to browse the labyrinth of of issues that exist. But again, the hope is that as we move forward, we can prepare ourselves and also learn from our mistakes over time. The current ethical issues at the moment, I think, from my perspective, is who gets to handle and use the data. And I think if we make it free, then hopefully we can have more regulations of how and when to use it, and we can be more savvy about it. At the moment, I think it's scary for me, because if we don't make it free, then as Siri said, only a few people have it. And what makes them so special, let's say, that we can't share that information with the rest of the world? Just because they have it doesn't mean that they're experts on it. And so it can go either way. You'll always have experts and people that will abuse it. The hope is that the more it's out there, the better we can generate a framework that protects ourselves and also does the best we can with the data we have without being too unethical. It's very vague an answer, but yeah, I don't know. Consta maybe has more insights as well.
I actually thought you put it really well, Kevin, especially about the regulations. Yeah, we need to talk and about regulations and what are the right regulations. I completely agree with you.
Okay, I don't have a, no, your answer, Kevin, was, I think, the best. But what I'm thinking is if we let the money, let's say, so companies have all this information and all this data can be used to make more money, then all these problems arise. So make it free, public, and can be used from everyone, but not for commercial, let's say, use. Because most of these data that we are talking of are samples from people with various diseases, or even just for different kinds of research in different kinds of genes in order to see mutations around the population. So if this data could be used against each one of us, then there is the crucial problem that we have to counter with legislations. And I don't know what it can be done, because in each country, even if you have big communities of countries that they share, let's say, the same rules, some countries will not abide by them. So still, we have to first, as a community, not only scientific community, but as the society, have to abide by some rules and then proceed to publishing everything and let everyone to be able to use it. So not following the money, let's say.
That is surely going to happen. And on that vaguely ominous note, I think I will start wrapping up. So yeah, this is our last in the series, and from my side, I would like to thank all our speakers for the time and the effort that they took to prepare their talks, and as well as all our supervisors for advice. I'd also like to thank our animator Dusan Knežević and his team for the amazing trailer that they put together. And most of all, I would like to thank Siri. It was great working with you to prepare this event. And thank you to everyone who came to the talks. I hope you learned something, and that you saw some cool scientific images, and that we managed at least a little bit to reignite your curiosity and remind you why fundamental research is important, and why evolution is really cool. And with that, I will leave you with Siri, who really did the majority of the heavy lifting in organizing this thing. And she will officially close the series. Siri, there you go.
Thank you. Well, I can just agree with everything you just said, and all your thank yous. I would also like to say a very big thank you to Milena, who is the yin to my yang. And also thanks to Francesca, our project manager of the Evocell network. And I would also like to say a big thank you to the speakers and an amazing network that we're all part of and that supports us. And I created this little collage to show you. Yes, so this would not be possible without all these amazing speakers, and as Pericles yesterday said, science is a team effort. So thank you to all of you who presented. And I would also say a big thank you to our funding source, the European Union's Horizon 2020 Research Innovation Program, as well as to the Museum für Naturkunde in Berlin for enabling us to host this event through their platform. And this event has been a very, very big learning experience for me, for Milena, as well as everyone that gave talks. I really hope this event, which was an experiment, I must say, it is the first time we ever done anything like this. We've not even done something like that in physical space. So it's, it's beyond a big learning experience. I hope this kind of event will inspire more of those sorts of science outreach and give you an insight into the speakers lab and work practices. And we hope we were able to spark your curiosity. And with that, I think I'd like to finish. I shared before in both chats the Evocell Network's webpage, where we will always update the newest information. We'll be developing a mini online exhibition next year. So through that webpage, you're definitely going to get informed, but also probably we'll post things through the Natural History Museum's platforms. And thank you so much.